Now the examples for the spore forming bacteria are the spores which are formed only by two genera of gram-positive rods, and these are of medical importance. There are so many bacteria responsible for forming the spores, but two important genera are important as a medically, that is Bacillus species and Clostridium species. Next is about endospore staining. So the detailed description of this endospore staining will be discussed later in the staining methods. So we can discuss briefly here. The endospore staining is a technique used in the bacteriology to identify the presence of endospores in a bacterial sample, which can be useful for classifying bacteria. So within the bacteria, these endospores are protective structures that we have discussed earlier, used to survive extreme conditions. But this protective nature makes them difficult for staining using normal techniques such as simple staining and gram staining. So they require special staining techniques. That are two important special staining techniques are Schiefer-Fulton and Moller stain. It is used majorly to detect and identify what the presence of a bacterial endospore and also a bacterial vegetative forms in a cell. And examples of this endospore forming bacteria we have discussed Clostridium species and Bacillus species. These bacteria naturally grow in soil, but they have great clinical implications in causing human bacterial infections. For example, Clostridium tetani causing tetanus, Clostridium botulinum producing a toxin called botulinum toxin, which present in the packed foods, that is packed meat food like sausages, etc. If it is contaminated with this organism, it produces this toxin and it causes paralysis by the intake of this contaminated or toxin produced meat or sausage. Next is Bacillus cereus. It also causes food poisoning such as vomiting, then stomach pain, etc., then nausea. And next is Bacillus anthracis. It causes anthrax in cattle and human. These are the examples of the bacteria producing endospores. These endospore staining techniques are classified based on the reagents used. So here I have already discussed that Schiefer-Fulton staining method and also Moller stain. So this method is also used here, mainly Schiefer-Fulton stain. In our laboratory or in the colleges, we usually do the endospore staining method using the Schiefer-Fulton procedure. And here the Schiefer-Fulton stain used is malachite green dye and saffron. In the donor method of endospore staining, this is another staining method for endospores, staining of the endospores. They use carbol fuchsin, acid alcohol and nitrosin solution. Moller stain is also there, that I will discuss later. So these are the methods. Application, this is simplified method. Application of malachite green, which is called primary stain. Then we are applying heat, act as a mordant for making it fixing. Then for the fixation process we are applying heat. Then we are application of the water, which acts as a decolorizer. Then we are applying saffron as a counter stain, and what happens? The green color shows the spores, endospores, and this pink color shows the vegetative cell. Because already while heating, what happens? This malachite green gain entry into the cell and it stains the endospores. That's why even though we are washing it with decolorizer with water, this endospore which have retained the green color is not going. It is not washing, that is a green color. Thank you. So these are all about endospores. Then the staining we can discuss in detail later.